70 years ago, science believed it was close to answering the deepest question in biology. How did the first living cell come to exist? Every generation of researchers said the same thing. Every generation of tools, the microscope, the sequencer, the cryo-electron microscope, revealed that the cell was more complex than the previous generation had imagined. This is not a story about what we know. It is a story about how precisely we have learned to measure what we do not. Chicago, May 1953. A 23-year-old graduate student named Stanley Miller sealed a mixture of gases inside a glass loop, ran electrical sparks through it, and waited. For days, nothing happened. Then the water began to darken. When he analyzed the residue, amino acids appeared on the chromatography paper, the building blocks of proteins, formed from nothing but chemistry and electricity. The New York Times called it a peek into creation. The scientific community called it proof. What neither noticed buried in the data would take 30 years to become a crisis. Miller's result was real. Amino acids formed. The chemistry worked, but every amino acid he produced came in two mirror image forms, left-handed and right-handed, in equal proportion. Life uses only the left-handed form. That detail was noted in 1953, recorded in the paper, and set aside. It did not seem like a problem. It was the problem. It would take the field 30 years to understand why. In the meantime, the experiment was celebrated. The building block problem appeared solved. The harder question, whether chemistry could produce the right building blocks in the right form, had not yet been asked. In 1984, Gerald Joyce and Leslie Orgel ran a precise experiment. They mixed small proportions of wrong-handed monomers into a reaction and watched what happened to chain growth. It did not slow. It stopped. The wrong-handed molecule introduces a geometric distortion the growing chain cannot recover from. One wrong-handed monomer, not many, one, and the chain terminates. In a prebiotic environment where both forms are always present in equal measure, that termination is not occasional. It is immediate. No polymer of biological length can form. The racemic result was not missed in 1953. It was understood and set aside. The field assumed homochirality was a downstream problem, something a later stage of chemical evolution would sort out naturally. That assumption lasted 30 years. What Joyce and Orgel demonstrated in 1984 was that it could not be deferred. Homochirality was not a problem for the next step. It was a precondition for any step at all. The field had known the fact since 1953. It had not understood what the fact meant. Every amino acid in every living cell is left-handed. Not predominantly, not mostly, exclusively. The ribosome that assembles proteins accepts only the left-handed form and stops dead when it encounters the other. Unguided chemistry, every experiment, every meteorite, every simulation produces both forms in equal measure. That gap between what chemistry does and what life requires is not a technical problem waiting for a clever solution. It is the first obstacle. And it has no demonstrated answer. Louis Pasteur discovered it in 1848, sorting tartrate crystals by hand under candlelight. He noticed the crystals came in two forms, mirror images of each other. He separated them, dissolved them separately. One solution rotated polarized light left, the other rotated it right. He ran into the corridor and embraced the first person he met. He had discovered molecular handedness, what chemists now call chirality. He had no idea, standing in that corridor, that he had just identified the founding asymmetry of all life on Earth. The ribosome is not indifferent to handedness. It was built around the left-handed form, and it enforces that geometry at every step of protein synthesis. When a right-handed amino acid arrives at the active site, it does not get incorporated slowly or inefficiently. It cannot react at all. The ribosome is a precision filter. The wrong-handed molecule fails the filter completely, and the chain stops. Chain terminator is not a metaphor. It is the mechanistic description of exactly what happens. Here is the problem stated at its most fundamental. Unguided chemistry cannot preferentially produce left-handed amino acids, not because the chemistry is insufficient, but because the laws of physics do not allow it. Fundamental chemical symmetry, the same physical laws governing every reaction in the universe, produces both mirror image forms in equal measure. No known natural process selects one hand over the other before life exists to do the selecting. The gap between what chemistry produces and what life requires is not a gap that more chemistry will close. Miller's experiment ran on a specific gas mixture, methane, ammonia, hydrogen, water vapor, because his advisor, Harold Urey, believed that was what the early Earth's atmosphere looked like. It seemed reasonable in 1953. 50 years of subsequent geochemistry revised it substantially. The early Earth's atmosphere was almost certainly nitrogen and carbon dioxide dominant, neutral to mildly reducing. Under those conditions, amino acid yields from spark discharge dropped dramatically. The experiment that appeared to solve the building block problem was run under conditions the early Earth likely never had. This is what the minimum viable living cell requires. Approximately 1,215 proteins, dozens of distinct functional systems, a ribosome containing over 50 structural components, and 3 RNA molecules, metabolic pathways, repair systems, membrane synthesis machinery, energy generation, and a genetic code fully implemented, not a complex organism. The floor. Below this level, the cell cannot replicate, cannot repair its DNA, cannot maintain its membrane. It ceases to function. This is what the origin of life field is trying to explain. Keep that in mind for everything that follows. By the time researchers could image the ribosome at atomic resolution, sequence minimal genomes, and reconstruct the last universal common ancestor from comparative genomics, the picture that emerged was not simpler than expected. It was more complex than anyone had imagined, extraordinarily integrated, with no known simpler precursor anywhere in the fossil or molecular record. The question the investigation must answer is not whether something this complex exists. It clearly does. The question is what preceded it. When Darwin published On the Origin of Species in 1859, he imagined the cell as something like a tiny blob of protoplasm, a simple, homogeneous unit from which complexity could gradually accumulate. He had every reason to think so. The electron microscope had not been invented. The genetic code had not been discovered. DNA's structure was 94 years away. Darwin's theory was built on a cell that molecular biology would later reveal to be nothing like what he had imagined. Each new instrument did not merely add detail to Darwin's picture, it replaced it. The electron microscope revealed internal architecture where Darwin had seen a blob. DNA revealed a digital information system where biology had assumed vague chemical inheritance. The genetic code revealed an arbitrary but universal translation table that no one had predicted. Cryo-electron microscopy exposed molecular machines of extraordinary precision. Each discovery compounded the previous one. The cell Darwin imagined has not been found anywhere. What has been found is something else entirely. A genetic archive written in a 4-letter digital code. a translation system that converts that code into proteins with 1 error per billion operations. A membrane maintained precise ion gradients, dozens of interlocking metabolic pathways, repair systems monitoring the genome continuously. This is not a complex organism. This is the floor, the minimum viable cell. Darwin imagined a blob. Molecular biology found a factory. The distance between those two descriptions is the origin of life problem, stated precisely. Darwin's theory of evolution by natural selection is not in dispute here. What is in dispute is the question that precedes it. Natural selection requires replication. Replication requires a cell. Before the first cell, there is nothing for natural selection to act on. Darwin knew his theory had a boundary. He drew it himself. In a letter to Joseph Hooker in 1863, he described the origin of life as a problem altogether too profound for human intellect, and he meant it. His theory of descent with modification was built to explain the tree, not the root. The origin of life is the root.
The origin of life is a problem entirely separate from my theory of evolution. My work explains how species change over time, not how life first began. That is a question for future science.
Evolution, but it begins with a cell already replicating, already generating heritable variation. The question of how that first cell came to exist is not a question evolution was designed to answer.
"1953, the answer seems so near. A spark inside a flask, and everybody cheered. Give us time, they said. The mystery's almost done, but every step ahead just showed another one. They found the code, the alphabet, the language in the cell. And suddenly the story wasn't simple anymore. Every breakthrough, every view, every microscope we built pulled another curtain back and showed a higher head. The deeper we look, the bigger it gets. The more that we learn, the more we haven't learned yet. Inside every cell, there's a city alive, a billion moving parts working all at the same time. The deeper we look, the harder it seems. The closer we get, the farther runs the dream. We thought we found the answer, but we're still asking why. The deeper we look, the mystery grows alive. Then came the Ribosome, a factory in motion, reading coded messages with microscopic precision. Proofreading, translating, building proteins one by one like a machine that somehow knew exactly what to become. And DNA kept whispering in every living thing, instructions nested in instructions like a song inside a string. Every molecule, every rule, every system that we find seems to point beyond itself to something in the design. The deeper we look, the bigger it gets. The more that we learn, the more we haven't learned yet. Inside every cell, there's a city alive, a billion moving parts working all mystery grows alive. And dimensions never seen. We're told that chance and time can somehow bridge the gap, but every year of science makes that story harder to map.
At the heart of the cell, the ribosome factory comes with activity. Here, tRNA molecules decode the mRNA, adding to the growing protein chain. This intricate process unfolds simultaneously across the cell.
Precision, more complexity than anybody told. The deeper we look, the brighter it burns. The more that we know, the more the question returns. Inside every cell, there's a universe alive, a masterpiece of systems working perfectly in time. The deeper we look, the higher it climbs. The more that the evidence refuses to unwind. We thought we'd reach the bottom, but the floor became the sky. The deeper we look, the deeper we look, the deeper we look, the mystery keeps replying. From the code to the ribosome, from the membrane to the mind. The deeper we look, the more we find.